Dear students, welcome to clear, correct and concise tips for MD path from R.A.T.S. Techniques in pathology for students. The topic for the day shall be immunofluorescence. As mentioned earlier, I shall be trying to balance the applied as well as the technical part of the topic. Think of the subheadings. The subheadings will take you to the answer. Whenever we do answer a question, we should be having a set of subheadings in mind. And these are what you people see on the screen. What is in the box is a vocabulary unique to immunology. Antigen is a substance which, when introduced into the body, leads to the production of antibodies. Whereas antibodies are immunoglobulins which are produced in reciprocation to the entry of antigens. Together they form the antigen-antibody complex, and the task of the day shall be visualization of this complex. There are different types of immunofluorescence. But first, let us see what exactly it is. This is a stage and in which I am having a material which can be the antigen or a proteinaceous substance, to which there is an exposure of the antibody which can be tagged to a fluorochrome or a bright substance. This substance is illuminated by means of a UV light or any other source of illumination, as we shall be seeing. And finally, the shine that is seen in the material is visualized. Generally, in a microscope, we see with the eyes, but here there are other modalities. So we shall be having this in mind. Coming to the types, one is direct. It uses a dye-conjugated antibody to stain the target protein. Direct immunofluorescence. Indirect immunofluorescence, on the contrary, involves the binding of the primary antibody to the target. And this primary antibody is detected by using a conjugation with a secondary antibody. So there are two steps, whereas in direct there is a single step. So please do remember there are two types of immunofluorescence and both of them are only technically different. Always I would like to touch upon a historical background. This was introduced by laymen way back in 1914 and it got developed into a fluorescent microscope by Bosch and Lohm in 1929. A histological staining of this was incorporated by Ettinger and Hedgeman stained the acid-fast bacilli. You people must be familiar with the Oramin rhodamin stain for the tubercle bacilli. And coming to the modes of the illumination, it can be biological, green fluorescent protein, GFP, or it can be because of organic dyes such as rhodamin, which I had mentioned some time back, fluorescein, cyanin, etc., or quantum dots. So these are some of the fluorophores or coloring material that could be used. And this is the original fluorescent microscope that was designed by Bosch and Lamb. It looks almost like an ordinary microscope to me, but then the hazards of directly looking into an immunofluorescence is beyond the dreams of today's lecture. A conventional microscope requires a much less light energy, 400 to 700 nanometers. And a fluorescent microscope, on the contrary, will require a much higher intensity. The fluorophores are the coloring material which we had spoken about. In a conventional microscope, there will be a source of illumination. It can be a mirror or a light, and through which the light source passes, traverses the material on the slide, and it reaches the eyes of the observer. Whereas in this, we are using three kinds of filters which I shall be talking about. There is a light source which passes through a filter and then it gets reflected onto the object, which is again reflected back. So this is the simple principle of the fluorescent microscope. This we shall be having in mind. I am very sure that you people will become familiar with this. And this is an easy diagram for us to draw. Again, the same thing is incorporated in a color diagram. Introduction, understanding, and drawing. So this is the objective lens. Objective lens is a series of lenses that helps in the convergence of a light to a point so that it creates a vision. So there is a light source. A UV light is one of the sources, but there can be other sources which I shall mention. There is an excitation filter, and then it turns back because of the dichroic filter, and it reaches the object which becomes colored or fluorescent. From here you find that there is a higher wavelength of light that is produced and it goes straight, and this is the emission filter through which it passes, and finally it goes to a detector or a camera. This is attached to a computer which displays the fluorescence. So I hope people are able to understand the diagram. As I told you, there are other light sources. A xenon arc lamp is mentioned over here. The ordinary LED lamp that is very famous now has been used. Lasers, mercury vapor lamp, UV light are all sources of light. And there are three filters: an excitation filter, a dichroic filter, and then an emission filter. There are three filters, please do remember them. And you find that there is a splitting of the light. It comes to the object through the objectives, and then it is reflected back at a higher wavelength which traverses dichroic filter and goes through the emission filter to the camera, and from there it is recorded onto the computer. I hope it is very simple. You will know the complexities later on. Look at this instrument over here. This bears all the description that I had been mentioning. Now, what are the general uses? General uses is not the application, but then we should have a beginning somewhere. It can be used to view small organisms or microbes. Also, three-dimensional clusters such as papillae or a three-dimensional cell or a tissue appearance. There can be antibodies which can be detected by means of the fluorescent tags. And there can be imaging of the genetic material, DNA and RNA. And finally, there is also some latest techniques such as the fish, which can be incorporated with the fluorescent microscope. So please do remember, small specimens, 3D structures, antibodies, DNA and RNA, and implementation of the fish. And this is the microscope that is bearing the complexities of the diagram that I had explained earlier. If you are not convinced with the complexities, please do see this. It is not worth going through. But then this amount of physics and precision is over here, and a lot of work has been done. I hope you people will be able to understand the intricacies of this. And the design of a lamp is like this. The fluorophore, it absorbs the light energy, the molecules get excited, they lose the absorbed energy. This is one of the disadvantages of immunofluorescence. It has got a very short half-life, as a result of which it becomes dimmer and dimmer, and maybe beyond 20 minutes or so it might not be fluorescent, and hence a photograph is mandatory for us to have a proof of the fluorescence. And there are two types. One is called an epifluorescent microscope, another is a confocal microscope. Please do remember these two types. And as I had mentioned earlier, it can be used for small samples to find out whether the tissue is viable or not, the DNA material, fish techniques, etc. This I am showing purposely the compound microscope that we are regularly using. You are finding there is a camera, or in this case there might be an eyepiece, and you find that there is a light source over here. There will be a series of objectives, and you find that there is a convergence of a light. There will be a prism over here which helps in exact focus of the light, creating a vision in our eyes. And this is the basic principle of it. So this I hope you people will be able to compare with the fluorescent microscope. What are the general advantages and disadvantages of a fluorescent microscope? I have put them in bold so that you people can note it down. Rapid tumor detection. One of the most important thing is a turnaround time. Sometimes we will have to make a rapid diagnosis. A graft rejection, for example, they cannot be waiting long. So a rapid tumor detection when the patient is on the table, or for a graft rejection, for a better nuclear visibility, to understand the tissue integrity or invasion. The plane of tissue can be chosen or changed, and there can be a morphological correspondence to the actual picture that we are seeing in the light microscope. To some extent we can imagine. So these are all some of the advantages. The disadvantages, the signals are qualitative. There might be a bright light or a dim light, but then a light is there. We will not be able to measure it. There is a lack of quantitative information. Photo bleaching or auto bleaching, it continues to lose its light, as a result of which beyond a time it is no longer existent. And it requires trained personnel who can handle the equipment, interpret, and who will also know the black and white images of it. So this is the list of advantages and disadvantages which we shall be having at the corner of our mind as we progress with the class for today. As I told you earlier, this is a technical class, and in a technical class I give more emphasis to the utility and the general applications. Coming to the applications, immunofluorescence can be used 1. in renal biopsies, 2. in cutaneous lesions, 3. in microbiology for identifying the organisms, 4. in neoplasia, and 5. for analyzing the DNA and RNA. So these are the general areas of usage. I'll first go to the renal tissue as such, and I'll give you a few examples. The first is a slide of a minimal change glomerulonephritis. So minimal change means there is hardly any change. It appears normal under the light microscopy. But then B is supposed to be the immunofluorescence, which is showing the IgM, and it is shown as granular deposits in the mesangium. So this is 1, minimal change disease. 2 is focal segmental glomerulosclerosis. Focal and segmental means some parts of the glomeruli or some of the glomeruli are affected, whereas if it is diffuse, then all the glomeruli and the entire part of the glomerulus can be affected. Focal segmental glomerulosclerosis, in which there is a deposition of IgM that could be made out. So I am not going to explain you people this, but these can be some of the uses of the fluorescent microscope in renal pathology. And the usage continues. Membranoproliferative. Membranoproliferative means the Bowman space is almost obliterated, and then there is a proliferation of the endothelium, the epithelium, mesangium, etc., filling up the entire glomerular space. And I find that there can be the C3 which can be deposited along the capillary walls. This is an IgA nephropathy which is showing again mesangial proliferation, which is eosinophilic. The darker ones are the capillary walls, and there can be deposition of IgA, so it can be called as IgA nephropathy itself. So look at the various usages. The difference in the pictures also you people can appreciate. Therefore, a person who is versatile in this will be able to identify. But generally it is advised that light microscopy, electron microscopy, then PAS and the silver stains along with immunofluorescence are mandatory in the interpretation of the renal biopsies, which I hope we shall be seeing some other day if time permits. And this is one thing I would like you people to kindly memorize for the time being. There can be primary glomerular lesions, there can be secondary glomerular nephritis, tubular interstitial nephritis, vascular diseases, as well as miscellaneous diseases. Initially, there is something pertaining to the nephrotic syndrome, then there can be immune complex or a crescentic glomerular nephritis, diffuse proliferative and a chronic glomerular nephritis. Only the conditions I have mentioned over here, not the distribution as such. I would like you people to kindly impart this in your paper when you are asked about the applications of the immunofluorescence. Renal immunofluorescence itself is a question which we shall be seeing later on. Secondary, there can be lupus nephritis in a case of SLE. Multiple changes are there. Amyloidosis, diabetic nephropathy, hemolytic uremic syndrome. Tubulointerstitial, obviously it is ATN. Acute tubular necrosis or tubulointerstitial nephritis. Vascular, hypertension, or sometimes polyarteritis also, and miscellaneous amyloid-like nephropathy. So all this can be identified. When I mention the word amyloid, I should have thioflavin T and thioflavin S in mind as special stains and immunofluorescence for amyloid. Graft rejection of the kidney is also another source where immunofluorescence can be applied. And the rejections are divided into hyperacute, acute cell mediated. Along with it goes the chronic. But chronic generally does not display much of immunofluorescent changes. And the corresponding antibody where they are exactly found are shown in the list over here. C4D, C4D again, C5b29 membrane lysis complex in a case of acute tubular necrosis, shock kidney, and C3 in a pure cellular rejection. So this particular couple of tables I would like you people to kindly memorize. So this is a projection of lupus nephritis in SLE, and I am seeing multiple capillary loops. After all, what is a glomerulus? A glomerulus is a set of tough capillaries in which there is a deposition of immunoglobulin G, IgG. And what happens is there is a coarse granular material that is deposited in the capillary loops. This could be made out in lupus nephritis. So these pictures are just to keep the lecture alive. As I mentioned earlier, there can be different applications. We had seen the renal application so far. What can be the applications in cancer? Immunofluorescence helps us to identify invasive cancers, high-grade tumors, to determine the margin status, particularly in the breast and the thyroid tumors, and in incorporating the fluorescent in situ hybridization, FISH. This has got its own uses. We will be coming to that at a later date. That is also a technique that has to be covered. Also, the list of uses continues to identify the interface nuclei, the transcription, and any other genetic abnormality such as chromosomal translocation. In the diagnosis of multiple myeloma and leukemia, in the selection of targeted therapy, and there can also be the usage of immunohistochemistry along with FISH for the detection of various gene proteins. So these are all the various list of applications in cancer. In one slide I have given you, I would like you people to kindly prepare a list and memorize. So renal applications we have seen. Applications in cancer we have seen just now. There are other uses. In SLE, which we had seen earlier, apart from the capillaries, there can also be the nuclear protein that can be displayed. Also, the leukemic cells can display immunofluorescence, and this also can be an attempt at making a diagnosis. Microbiology is a close cousin of ours. In fact, we got separated from them. And immunofluorescence is used for many a diagnosis, such as trichophyton or yeast, gram-negative bacteria, antigen antibody in the tissues, such as even hepatitis, vesiculobullous diseases of the skin and oral mucosa. These are for autoimmune diseases. And to titrate the antibodies against the antigen and also the autoantibodies. These are all the various things that could be diagnosed. This incidentally is a Legionella bacilli that has been given a fluorescent tag we are able to identify. Such will be the color also in a case of Oramin rhodamin stain for tubercle bacilli. The same picture, for the sake of repetition I am doing it. Specimen over here, series of objectives. This is the source. There is an excitation filter, dichroic filter or mirror, and then an emission filter. Finally, it goes to the detector, that is the camera, and finally it is fed on to the computer. This diagram you people can draw in your examination. And this again helps in defining the two types. One, there is a tag that is already attached to the antibody gets attached to the antigen. This is called a direct immunofluorescence, whereas an antibody to the antibody is attached, so this becomes indirect, and the secondary antibody, it has got a tag to it. So the two types you people will be able to mention in your answers. The next series will be dermatology. Dermatology, you find that it is a nightmare for us. Making diagnosis of vesiculobullous diseases without immunofluorescence is a real nightmare. And thanks to this picture, it is from a particular source that I had mentioned over here. It can be used for many an autoimmune disease. I would like you people to kindly go through the entire list that I am projecting and by heart it. So, in bullous diseases such as pemphigus, pemphigoid lesions, herpes gestations, and dermatitis herpetiformis, linear IgA bullous diseases, all this can be used. Connective tissue diseases such as discoid lupus and systemic lupus erythematosus, vascular diseases, allergic vasculitis, hinox-conolin purpura, polyarthritis, and other diseases such as porphyria, lichen planus, connective tissue disorders such as systemic sclerosis, psoriasis, dermatomyositis. So an entire tag is given and you people will have to divide this. There can be bullous, connective tissue, vascular, other, and connective tissue again. So I would like you people to kindly memorize it. And you need not give the entire list of examples. You can choose examples of your choice. And these are some of the antibodies which are used in dermatopathology. You can go through them and the conditions are given over here. Antinuclear antibodies, for example, are positive in 90% of the cases of SLE. And these are all the other antibodies used in other conditions. So, I would like you people to kindly explain this. This is the nth time I am showing this picture again. Light source, excitation filter, dichroic mirror, and it goes on to the object, it becomes fluorescent. Then there is an emission of a higher wavelength, it passes through all the filters and then goes to the detector. And the subject that can be used over here is fluorescent isothiocyanate. That is over here. So this is a primary antibody, there is a secondary antibody, this is a label. And what is that label called? Fluorescent isothiocyanate. Please do mention that. What is the coloring substrate? You will have to mention. Earlier I have forgotten to mention what can be the mode of transport. It is not formalin that we will be putting it in. We will have to do it in a frozen section. So send it as it is, a frozen section, a thin section is cut, after which we will be proceeding with the staining. Now, dear students, As we are nearing the end of the class, I would like you people to kindly concentrate on my repeated description to this. There is an LED light over here. There is an excitation filter. You find that the light energy is passing through a lens and then a dichroic mirror. It is getting reflected, it goes on to the object that is kept on the stage. And after it becomes fluorescent, you find that light energy of a higher wavelength is produced. It passes through the emission filter and it goes on to the camera. From the camera, you find that it is fed on to the computer. This will be the identical principle even in an electron microscopy at a later date. I will be explaining. So, step by step, I would like you people to explain. Now, what is this word 2D? 2D means it is just two-dimensional. That means there is something else. So, what is the next thing that I would like to mention? Now, coming to the other applications of the immunofluorescence. I would like you people to kindly appreciate this beautiful picture over here. So long we have been talking about either green lines or blue lines, but here I am finding a myriad of colors. So there is an orange, a light green, a blue color, etc. And this is the nucleus per se, in which you will be having material such as tubulin protein. And then there can be other material also which are indicating the mitochondria, enzyme sirtuin 6. These are all names which the company will be having so that they will be maintaining the privacy of it. Otherwise, tomorrow you and I will join together and be manufacturing it. And courtesy to Nancy Kedersha, and this is a picture of an osteosarcoma. See how all it is being used. And the three-dimensional also. You find that here there is, it looks like a two-dimensional papillae, or it has got a finger-like tuft. But then it is more obvious in these, and a three-dimensional picture can also be brought about by means of this immunofluorescence microscopy. This is something that you people will have to explain the various sources, then the sample that is being placed, the tag that is being attached to the antibody and being visualized. I would like you people to compare this particular picture, which is a superb line diagram, with this. So here it is an indirect immunofluorescence. The same picture is over here. This is literally kept over here. So there is an antigen or a protein to which the antibody is getting attached. And instead of a tag over here, it has got provision for attachment of another antibody. This is called a secondary antibody. This secondary antibody has got the fluorescent material along with it. After which there is a fluorescence which will be found emitting from the mounted specimen. And this is seen on the screen over here, which will be recorded. Always a recording is mandatory. Even an MRI you people would have seen. It is not just one picture that is final. They edit and finally they choose the best out of it. This is just a line diagram I would like you people to kindly explain. In this series, there is an antigen that is present. Here there is no antigen. So when an antibody is trying to be attached to it, this gets attached and ultimately it can be visualized by means of an UV light. But here, since it is not there, there is no attachment, it gets removed, and finally when there is a UV light, you find that there is no fluorescence. I hope this makes matter very simple. Whether it is going to be ELISA or immunohistochemistry, this will be the basic principle which we shall be following. Where there is no vision, there is no hope. I hope you people have understood this and kindly rehearse. Repetition is the mother of knowledge. Thank you.